My name is Kevin Lafferty. I'm a marine biologist with the U.S. Geological Survey, and I live here in Santa Barbara, California. Behind me is the Pacific Ocean, and I study the Pacific Ocean and the animals that live in it. In particular, I study the parasites that infect those animals. Now, you might not be surprised to know that just like all animals on land, all animals in the ocean and lakes and streams and rivers, they have parasites too. In fact, this is where most parasites probably evolved. They've been around for a long time, so they've had a lot of opportunity to evolve incredible strategies for infecting marine and aquatic organisms. Now, this ocean behind me has a big kelp forest, you can see that, and also out beyond into the Santa Barbara Channel, there's a complex food web, and parasites are a big part of that food web. At the bottom of the food web, we have small plants called phytoplankton, and those phytoplankton have their own parasites. They have fungal parasites, they have bacterial parasites, they have viruses that infect them. As you move on up for the food chain, we find parasites at every stage. In fact, parasites comprise more links in that kelp forest food web behind me than the free-living species do. There's just as many parasite species out in that kelp forest as there are fishes and invertebrates and seals and sea lions. Now, thinking about the food web, we were recently doing an expedition out in the Santa Barbara Channel, and we came across what we think is the top of the Santa Barbara Channel food web. We stumbled across a dead humpback whale, a whale perhaps that had been struck by a ship. We pulled up next to this whale, and crawling on the surface of the whale are parasites. One of the most common parasites on the skin of whales are these amphipod parasites. They're crustaceans. They're related to free-living amphipods that are common in the kelp forest out here, but they live on the skin of the whale. They feed off some of the decaying skin. They probably don't bug the whale that much, just like as if there was a little parasite feeding off your dead skin on your body. And then also on the humpback whale are these barnacles that live embedded in the whale tissue. Now, these barnacles are not feeding on the whale per se, but they use the whale as habitat, and they're highly adapted to living on whales. Now, what's interesting is that some whales, or some barnacles have actually figured out how to feed on their hosts. And a great example of that are barnacles called Rhizocephaline Barnacles. And these barnacles generally infect crabs and other crustaceans. They settle on the crab just like a normal barnacle larva would. But unlike a normal barnacle larva, which would then start to form a shell and feed on plankton, the parasitic barnacles inject themselves into the crab with a hypodermic needle that they make themselves. And these parasitic barnacle cells float around in the crab, they start dividing, and they begin to build a fungal-like network that branches throughout the crab's body, and it starts absorbing nutrition directly from the crab. The barnacle that's inside the crab doesn't take all the crab's nutrition. It wants to keep the crab alive, and that's a general feature of most parasites. Parasites are happy when their hosts are happy. And so the barnacle doesn't take all the nutrition, just takes enough to keep the barnacle growing and starting to develop, but it doesn't steal enough nutrition from the crab that it hurts the crab. Now, the barnacle eventually gets big enough that it wants to produce its own eggs, and the perfect place for a barnacle to put its eggs is underneath the tail of the crab. Female crabs have big, wide tails that they put their egg masses under, and so that tail is perfectly evolved to support and shelter eggs. But if the barnacle happens to find itself in a male crab, it doesn't despair. Instead, what it does is it manipulates the hormones of that male crab and turns it into a female-looking crab. It makes that male have a wide tail so that barnacle can put its own eggs underneath the male's abdomen. Not only that, it changes the behavior of the male crab into more female-like behavior, because when the barnacle finally puts its eggs underneath that crab's tail, the crab takes care of the eggs for the barnacle. thinking that the barnacle's eggs are its own. And so this barnacle has a very, very heavily evolved relationship with the crab, something that we call parasitic castration. It takes all the extra energy that the crab has, that the crab would normally put into reproduction, it takes that energy for its own, and instead of producing crab babies, well, the crab produces barnacle babies instead. Now, there are lots of parasitic castrators out in the marine environment, and one that I've worked with a lot is called a trematode. Trematodes, as adults, live in the guts of vertebrates in the marine system. They would live in the shorebirds that are flying around behind me. But the eggs of the trematodes come out with the feces of the bird, and they go on to the intertidal zone where lots of snails are crawling around. They infect those snails, and the larval worms crawl into the gonad of the snail. They live there, and they feed on the gonad, and they reduce that gonad to nothing. Once they've filled up the gonad, that snail will no longer produce baby snails. Instead, it produces parasite larvae. Now, those larvae go out into the environment, they swim around, and they look for the next host in their life cycle. And that host is oftentimes a fish. And the most common parasite in our area infects one of the most common fish in our area, and that is called the killifish. Killifish is a small fish. It's a few centimeters long, and the parasite migrates through the gills of the fish up into the brain of the fish and begins to coat the surface of the fish's brain with small baby parasites. So we wondered, why coat the surface of a fish's brain? One of the reasons we hypothesized was because the next host in the life cycle was to get back into that bird where the adults lived. And so perhaps by coating the surface of a fish's brain, the parasite might be in the driver's seat. Specifically, it might be able to manipulate the fish's behavior in a way that would make it easier for a bird to catch that fish and eat it. I had an undergraduate working with me at the time. His name was Kimo Morris, and he really wanted to work on this for a project. And so he set up an experiment where we caught a bunch of fish, we put them in an aquarium, and then Kimo, my student, watched those fish swim around, and he looked for certain behaviors that made the fish more conspicuous to him, because he figured that if those fish attracted his attention, maybe they would be attracting the attention of a bird. And he did notice that some of the fish did some unusual things. They would swim up to the surface, they would roll over on their side, they would scratch on the bottom, etc. And so he kept track of those, and he gave them specific names. And then he watched individual fish for a half an hour. He wrote down the unusual behaviors that they did, and then he caught them and he dissected them and counted the number of parasites that were on those fish's brains. And what he found was that infected fish were 4 times more likely to exhibit these unusual behaviors compared to uninfected fish. And that made him wonder, hey, maybe these infected fish are easier for birds to catch. And so Kimo set up a field experiment where he put fishes out into a lagoon. He put some into a cage that was protected from birds and some into a cage that was left open and allowed birds to come in and feed. And he went and he put some uninfected fish and some infected fish in each cage, and then watched those cages for a few days. And the first day, no birds came and fed in the cages. And the second day, no birds came either. And just about the time he was going to give up, a great blue heron stepped into the open cage, and it ate a fish, and then it ate another fish. And after a couple of days, Kimo brought all the fish in, he dissected them all, and he compared the fish that were in the protected cage where no birds were feeding from the fish that were in the cage where the birds were allowed to feed. And by counting the parasites on the brains of the fishes in the different cages, what Kimo was able to find is that very few uninfected fish were eaten by birds, but almost all the heavily infected fish were eaten by birds. And the only way that this could happen is if infected fish were 10 to 30 times more likely to be eaten by birds compared to uninfected fish. And this was a very strong indication that parasites are able to manipulate the behavior of their hosts, but not only that, that they're actually involved in driving the food web. In other words, it may be possible that these parasites being present in the environment makes it easier for birds to catch fish and actually determines who lives and who dies in that fish population. Trematodes are not the only parasite that's able to manipulate the behavior of their host. I was working with some Japanese colleagues, and they were trying to understand how to recover an endangered species of fish called the Kirakuchi char. They did a lot of amazing detailed studies on these char. For instance, they would tag them, they'd visit them every month, collect them, look inside their stomachs to see what the char were eating, put them back in the water, weigh them, measure them, etc. And one of the things that they noticed was that most of the time, char eat insects that fall in from the forest canopy or insects that are living in the stream bed, aquatic insects. But during the months of September and October, the stomachs of the char were filled with crickets. Now, crickets usually are not associated with streams, and they don't have any business being in the water. And so this raised a lot of questions for them. Why are the trout stomachs full of crickets? Well, with further study, what they were able to deduce is that there was a parasitic worm, a worm called a nematomorph worm, that was infecting these crickets. And these worms are well known to manipulate the behavior of crickets in such a way that it causes the crickets to jump into water. The reason that the nematomorph worm wants the cricket to jump in the water is because the adult worm will live out its life mating in a stream to make parasite eggs that will then go and enter into crickets down later in the worm's life cycle. So to get the cricket to do something crickets don't want to do, which is to jump into the water, the nematomorph has some abilities to manipulate the behavior of the cricket. Now, what's interesting about this case in Japan was not only was it an observation of parasites manipulating the behavior of a host, but also that manipulation has great consequences for the food web in those streams. Specifically, when my Japanese colleagues tallied up all the calories that trout ate through an entire year, they found that crickets provided more than half of those calories. And those calories were delivered to the trout by a parasite. Furthermore, because the trout were feeding on crickets instead of their normal food, that changed the nature of the stream food web. So in other words, the trout were full of crickets. They weren't feeding on the insects that live on the bottom. That meant there were more insects living in the stream. Those insects were more common. They did insect things like shred up leaves and detritus. They fed on the algae that grew on the rocks. That changed the amount of flow of nitrogen that moved through the stream, etc. So here another example of a parasite that's involved in a food web and has some pretty striking impacts on it. Not only that, but by providing more than half the calories of this endangered species, that endangered species might even depend on the presence of that parasite to persist in Japan. One of my favorite parasites that we see is called Herudonella. And it's my favorite because it really is such a romantic story. When we go deep sea fishing, we haul up oftentimes these fish called wahoo. And they're a lot of fun to catch, but they're even more fun to cut open and look inside their stomachs, because inside the stomach of most of the wahoo that we catch are these large thumb-sized worms. And not only are they big and active and they suck onto your hands and stuff, but they live together, usually a pair. They're embracing each other almost for their entire lives, and they do nothing but make love to each other and suck blood from the stomach of their hosts. It truly is aspirational in my mind.
that just filled with
Blood.
fish blood?
Yeah.
Oh, you probably like that kind of food.
It's pretty black. You know, what's the thing is, these things are going to smell so good. So how many pieces do we have? 1, 2, 3, 4, 5, 6, 7. Do we have any garnish? Look at that one, you got the sucker right there.
See the sucker on that one?
Actually, it's not bad, John. What do you think? It's not terrible.
The reason we're looking at fish in coral atolls is we're curious about how fishing affects parasite communities. Now, you might think that when we go in and fish on a coral reef that we disturb the environment and that creates lots of sick fish, so we would have more parasites. But in fact, our hypothesis was just the opposite. That as we fish a coral reef system, we simplify the food web, and that makes it harder for parasites to complete their life cycles. Their fish hosts might not be as abundant, they might be missing some of the hosts in their life cycle, and so what we would expect is that areas that are heavily fished have fewer parasites than areas that are not fished. Well, it turns out it's pretty easy to find heavily fished coral reefs. Most parts of the world where people live on coral reefs, the people there depend on fish as a main source of protein because many of them are quite poor. And so those reefs are heavily fished. It's a lot harder to find places that aren't fished. And to do that, we go to a special place called Palmyra Atoll. Palmyra Atoll is a nature reserve, and it's managed by the Nature Conservancy. And it has a research station, but no indigenous human population at all. And so the reefs are unfished, and in fact, the reefs are protected from fishing. Once you land in Palmyra Atoll, you see this beautiful environment, and the first thing you want to do is jump in the water and go snorkeling and look at the fish. And when you do, the fish look back at you, and they appear terrified. And one of the reasons that they're so nervous is because what they see most of the day are these large predators swimming around. You see, in an unfished coral reef, predators are protected, and so they become quite abundant, and that changes the food web a lot. And that also makes new opportunities for parasites in these food webs. Well, our idea was to go to fished coral reefs and unfished coral reefs, sample both, and look at the parasites inside 5 different species of fish. Here are the different species, and the idea was to go through and sample these guys by catching them with nets and then bringing them up to the lab and dissecting them. What we found was that we couldn't catch them with nets, and so instead we moved to trying to shoot them with little micro spears. And that worked pretty well, except for the fact that with so many predators around, particularly these large gray reef sharks, we oftentimes would set up shark feeding frenzies, particularly when we tried to spear the larger snapper species that we were interested in. So to get the snapper, we switched to hook and line. And even then, sometimes when we pulled up a fish on the end of a fishing pole, what we found is that the shark had had its way with it first. When we looked inside those different species of fish, we found many parasites that had never been seen before. And then we compared those to the parasites that were found in the heavily fished area. And we found that for each species, there was a higher richness of parasites in the unfished Palmyra Atoll compared to the fish sites. And my colleague Chelsea Wood has traveled around to many other sites in the Pacific repeating these studies and found even more interesting relationships between fishing and parasites in the marine environment. Now, I don't mean to imply that parasites are good. Parasites are neither good nor bad, but sometimes they certainly cause problems, and that can happen in the marine environment just as much as anywhere else. When I was younger, just a new marine biologist, one of the things that I like to do is go down at low tide and explore tide pools. And one of the most common species in those tide pools was a snail called the Black Abalone. And they were so abundant that sometimes they would have to sit one on top of the other to find space. Now, we don't know exactly when this happened or where this came from, but a new mysterious disease arrived sometime in the mid-'80s. And people that had been tracking abalone watched as their populations crashed from one site to the other, from hundreds of individuals to 1 or 0 individuals. This completely changed the nature of the Rocky Intertidal Zone. These were one of the dominant species, and really in a matter of years they were gone. And this continued year after year as the range of the abalone began to collapse in the southern area of California to the point where it appeared that that species might go extinct. In fact, the abalone was listed as an endangered species directly because of this particular disease. Now, it took a long time to figure out why abalone were declining. There was a shellfish pathologist who did a lot of histology on sick and dying abalone. Her name was Carolyn Friedman, and what she discovered was that these abalone were infected with an intracellular bacterium called a rickettsia. And this rickettsia would go into the digestive gland, and it would start to kill the cells that the abalone needed to digest its food, which is drift kelp that comes in that the abalone feeds on. And unable to digest its food, the abalone would start instead digesting its own muscle tissue. The foot of the abalone would then shrivel up, and when that foot shrank down to a small enough size, it wasn't able to hold on to the rocks. And the next storm that came through would knock it off the rocks, and then the seagulls would come and feast on the remains. The near extinction of this species by a parasite is quite unusual. In fact, it was so unusual that many of us wonder if perhaps that parasite came from elsewhere. Abalone are distributed around the world, and they have different types of parasites, and we've known in other cases that when a new parasite comes in from outside, the animals that live in that area are exposed to a parasite that they don't have any evolutionary adaptations to, and that can be quite problematic for them and lead to high mortality. Now, the story on the black abalone isn't finished. We've lost many of, or most of the range of the black abalone, but recently Carolyn Friedman discovered what looked to be a new parasite. And not only did it look to be a new parasite, it didn't appear to be killing the black abalone at all. But when she looked closer, what she discovered was that this new parasite was actually an infected version of the old parasite. In other words, this parasite that was killing the abalone got its own parasite. Specifically, it got a viral phage that came in and infected it. And those infected parasites were not as damaging to abalone as the uninfected parasites were. And so this addition of a new parasite to the old parasite benefited the original host. In recent years, we've seen more and more stories like the black abalone story. Coral bleaching with indication that infectious agents are causing corals to die around the world, or a massive sea star die-off with suggestions that perhaps there's some sort of virus or bacterial pathogen that is killing sea stars up and down the coast. Sea lions that are dying from all sorts of infectious diseases, sometimes that cause malnutrition. And there's lots of examples of other parasites of concern for animals that we care about. And so in those situations, it's really important to understand what these new types of parasites are. And in the ocean, that can be quite challenging because we don't have a lot of expertise in marine parasites. In other words, most people that study parasites of humans or animals or the crop plants that we depend on. And fewer marine biologists have been trained in how to recognize and understand the role of parasites in marine systems. But overall, I think that parasites are clearly natural parts of the ocean, lakes, and streams around us. They have fascinating, highly evolved life cycles. They're neither good nor bad. They are simply part of nature's complicated web and circle of life. Well, back outside in Santa Barbara, and I wanted to return to our story with the dead whale. I was out there with my friend checking out the whale's barnacle and amphipod parasites. We noticed something out of the corner of our eyes, and my friend yelled at me and said, Kevin, check that out." And I turned and I looked, and I saw something that I always wanted to see and never saw before, which was the fin of a great white shark. That shark had been feeding on the whale, feasting on it, in fact. And when we came up, put our boat next to it, it wanted to let us know that it was time to move away, and so it came for a better look. And as that shark came, I was able to lower my camera into the water, watch it approach. It turned, and I looked it in the eye, and it looked at me in the eye. And I thought, now I've finally seen the top of the food web in the Santa Barbara Channel. But later, when I looked at the video, what I realized was that the shark wasn't actually the top of the food web. In fact, if you look at the dorsal fin, you can see these long, trailing white threads. Those are the egg strings of parasitic copepods that feed on great white sharks. And they're the real top of the ocean food web. So I'd like you to think about that next time you're looking at the ocean and imagining the incredible food web there, who the real top of the food web is, and it's probably parasites of things like great white sharks. Well, I've really enjoyed telling you about some of the work that we've done on marine parasites and aquatic parasites. I hope that you'll see the ocean differently, and I think it's time for me to go out and go surfing.
Good morning, afternoon, or evening, wherever you are in the world, and thank you so much for watching this video. I'm Chelsea Wood. I'm a parasite ecologist at the University of Washington in Seattle. In Shakespeare's The Tempest, one of the central characters, Antonio, is contemplating committing a murder when he speaks the following line: "What's past is prologue, what to come, in yours and my discharge." Today, I'm going to talk to you about my very favorite topic, which is, of course, parasites of ocean ecosystems. I'll tell you what we've learned about their past, what that means for the future of marine disease, and what we should do in response. I don't mean to imply here that murder is the only reasonable course of action, but the things that we will need to do are new and different, and perhaps a little bit counterintuitive. Now, I started in on this line of research because there's one question that really drives my interest in marine biology, and it is, what did the oceans look like before people? If you spend time on the water, you probably have some intuition that marine ecosystems look a lot different today than they did in the past. That intuition isn't just nostalgia. There are hard data to suggest that the oceans have changed substantially in the past decades, and that people are primarily to blame for this. One of the easiest ways to see how human impacts reshape fish communities is to think about what a trophy fish was just a generation ago. This is a set of pictures from a 2009 paper of Lauren McClenahan's. Lauren went into a public library in Key West and she found these old trophy photos taken of the same hanging board on the same wharf over the course of 60 years. These photos reflect and document the state of Key West's reefs over a long time period, and there are a number of patterns that we can pick out in these pictures. Over time, fish become fewer, they become smaller, and we see many fewer predators, which fishermen love to catch. As a consequence of that reduction in predators, we can sometimes see an increase in the abundance of the prey species that those predators target because they flourish in the absence of anyone who eats them. This is just one particularly visual example, but of course it's a global phenomenon. And historical work gives us a pretty good idea of what the oceans used to look like before these impacts accumulated, at least for many of these large charismatic species. But what's going on under the surface? Each fish here is an ecosystem in itself, and that internal ecosystem is typically concealed. But what if we could unveil it? What sorts of parasites would we see in this scene? Were there more parasites in a pristine ocean of the past, or fewer, or the same overall number but a different mix of species? The problem is that we have literally no idea. There are almost no data to answer this question, and that's terrible because the answer is not obvious. We can make reasonable hypotheses that predict very different outcomes. It's easy to imagine how human impacts might increase or decrease parasite abundance, depending on the effects on the fish hosts. Here, we'll use the same time series of dayboat trophies in Key West. I'm going to layer on some dots to represent fish parasites, and this conceptual diagram will illustrate what's happening in the photos. Human impacts are increasing as we move through time and across the x-axis. Now, human interference doesn't reduce the density of every single fish species. For example, fishermen like to catch big predators like sharks, and that's good news for the prey species that get eaten by sharks because they can flourish when no one is around to eat them. If this prey host is increasing in density, we might expect to see a matching increase in the abundance of its parasites. In the disease ecology literature, this would be called the dilution effect hypothesis. But it's also easy to imagine how we might see the opposite pattern, and that's because hosts are both habitat and resources for parasites. Parasites may become less abundant as human impacts accumulate because those impacts reduce fish density or size or the complexity of food webs, all of which should reduce opportunities for parasites to be transmitted. We'll call this the healthy ecosystems are rich in parasites hypothesis, after a tree paper that was co-authored by Kevin Lafferty, who you just heard from. But of course, when I say the word parasite, I'm invoking this sprawling group of organisms who are diverse in terms of their bodies and lifestyles and habits, and there's no reason to expect them all to do the same thing. So we might also expect a mixed response of parasite abundance to human impacts. Here I've changed the colors of these dots just to indicate what line they correspond to in the conceptual diagram. And this begs a big and important question, which is, how do human impacts affect whole parasite assemblages? What parasites win consistent with the dilution effect? And then are there any that lose consistent with the healthy ecosystems are rich in parasites hypothesis? The problem with addressing this question is that, as I mentioned before, there are almost no existing data that tell us anything about the abundance of marine parasites in the past. And that's why my team started a project that we call Parasites of the Past. In this project, we circumvent the whole problem with not having any historical data about marine parasites by going straight to the source. Natural history museums have basements full of old animals in jars. When these animals are dunked in ethanol or formalin, their parasites are preserved right alongside them, which means that we can find out exactly how many parasites were infecting that host at the time of its death, whether that death was last year or 100 years ago. So my team is tackling the problem of decoding the history of marine parasitism by performing parasitological dissections of museum specimens across 8 fish species. These fish species were chosen to span a variety of body sizes, and we've got some predators as well as some fish species that are very low on the food chain. These fish in jars are literally parasite time capsules passed down to us from decades ago. We're just finishing the data collection for this project, and I don't have the data from all 8 host species to show you, but I can show you the data from the very first host species of the project, this guy shown here, English sole. I want to begin the results by getting you oriented to one panel of what will ultimately be a multi-panel plot. What I'm going to show you are a bunch of time series spanning from about 1930 to 2016. On the horizontal axis will be year, and then on the vertical axis is parasite abundance. The pattern of change through time will look like this, and each plot will represent a single parasite species. I'll indicate to you the taxonomic group of the parasite with these cartoons. We'll have copepods and leeches, as well as trematodes, nematodes, cestodes, and acanthocephalans. And I've grouped up these parasites by their responses. In total, we detected 12 parasite taxa in this one host species, English sole. Over the 85 years of the dataset, 9 of those 12 parasite taxa did not change in abundance. 1 of the 12 species increased in abundance through time. This is a trematode larva called a metacercaria that we detected far more of in recent years than in the deep past. And 2 of 12 species have actually declined over time, sometimes becoming so rare that they were hardly even detected in the more recent specimens. So when we look across all of the parasites in one host species, we see this whole universe of possibilities. Most parasites stay pretty steady, some just take off in abundance, and some dwindle away to nearly nothing. This suggests to me that as we move forward into the future, thinking about what's going on under the surface of this visible world among the parasites who are typically hidden, that we're not necessarily in for an explosion of marine disease, but neither are we in for a wholesale loss of parasite species. Instead, we're going to see both of these outcomes simultaneously, with some parasite species being winners in this changing ocean and others being losers. I can't say with a ton of certainty at this point which are the parasite taxa that are likely to increase and which ones are likely to decline. But I do know that we're going to see both of those things happening as we move into the future. And both of those outcomes have important implications for our ability to feed the world with seafood and to rely on functioning coastal zones for ecosystem services. And that's because the losers probably include lots of parasites with important roles to play. That might sound counterintuitive, but as you just heard from Kevin Lafferty, parasites are natural parts of ecosystems and they perform vital ecosystem functions. We can talk more about this later on, but to keep it brief, they might keep pest host populations in check. They might lubricate vital predator-prey interactions. They might actually be responsible for subsidizing apex predators. And losing these parasites could mean losing the ecosystem services that they provide. Meanwhile, the winners probably include parasites that we don't want around. And these include pests of aquaculture and wildlife, maybe even pests of people. What's past is prologue, and by that, Shakespeare meant everything that has happened in the past has led us to this pivotal moment when we can choose one path or another. We're faced with two challenges. On one hand, we've got this rising tide of marine disease parasites that are winning as we mess with the world's oceans. And on the other hand, other parasites are disappearing into extinction, often before they can even be named. Someday, the only place they may still exist will be in jars. What do we do about that? That, my friends, is in yours and my discharge. Thank you.
Hey everyone, welcome back. So if you have any questions for Dr. Chelsea Wood and Dr. Kevin Lafferty, please feel free to post them in our YouTube chat. And hello, Dr. Chelsea Wood. Thank you for joining us.
My pleasure.
Okay. Let me post our very first question to you here in the chat. So our very first question to Chelsea is that, were all the fish from the museum collection coming from the same area of the world? If yes, do you access to the same fish species from different places in the world to compare the diversity of the parasites found?
Great question. Yeah, so in the data that I presented just a minute ago, the museum specimens all were collected from Puget Sound, which is an inlet in Washington State that is adjacent to where I work at the University of Washington. We decided to focus on just one region because there can be so much variability in parasite communities, and we wanted to just focus on the variability over time in a single location. The data that I showed you were for one host species, English sole, but I'm really excited to report that we now have the data from all 8 of the host species that we were able to dissect, and soon we'll be revealing the results from those analyses in presentations and papers, and the stuff that we're finding is really cool. So we've reconstructed basically like a century of parasitological change for this one region of the world, Puget Sound. But of course, we're really excited to see what's going on in other places, in part because this is a super powerful method for understanding the parasitological history of a location. And museums have tons of specimens, so we're not really limited. We can go to many different locations, and that's what we plan to do.
Thank you. For our next question, Dr. Korsk asked, looking beyond museum collections, are there any other sources of parasitological data you feel are underexploited?
Definitely. Yeah. So we also sometimes use old data sets. These are really hard to come by. I have been scouring depositories of data, people's old field notebooks, querying colleagues, looking through the literature for historical parasitological data sets for the past decade. And I found 2 that are usable. There aren't that many. In order to use a parasitological data set, we need information on the size of the host that was collected. And often you'll get parasitological data from the past, but have no idea about the size of the host. And then it's really hard to know whether you're actually seeing change in parasite abundance over time or merely a change in the body size of the host, which can have important impacts on the abundance of parasites. So we have been looking to these old historical records of parasites, but there are some other options as well. Ancient DNA is a technique for resurrecting the DNA of any organism, not just parasites, but anything else. From, for example, fossils, archaeological remains, coprolites, old sediments. And I don't do any molecular work, but I have lots of colleagues who are able to pull parasite DNA out of specimens that are quite old. Ancient DNA is the reason why we know so much about the medieval plagues. We're able to pull the DNA of Yersinia pestis, the bacterium that causes plague, out of the root tissues of teeth of plague victims that have been preserved in crypts in Europe. So ancient DNA is a really cool way to learn about not just ancient parasites, but even parasites that have existed contemporarily or in the past couple of 100 years. And then one other thing, you asked beyond museum collections, but just looking beyond liquid preserved fishes in museum collections, there are lots of mammal and bird specimens where all the viscera have been removed, right? When you preserve a mammal or a bird for use in a museum, it's usually just the skin. But the ectoparasites of those skins persist, and you can actually comb the skins of mammals or the feathers of birds and pull their ectoparasites out, even if they've been prepped for presentation in a museum. So there are lots and lots of ways of getting at this historical parasite abundance, and many of them are quite underutilized. I feel like it's super low-hanging fruit if there are young scientists out there casting around for a parasitological project to work on. I highly recommend this avenue because there's still so much to be discovered.
Thank you, Chelsea. For our next question, Miriam Kelly asked, do you plan to expand your research to see where else this could have most likely occurred, like outside North America?
Yeah. One goal that I have in the long term is to get into some European museums. This will be interesting for a couple of reasons. One is that European collections tend to be quite a bit older than North American collections. I'm super lucky to be at the University of Washington, where we have literally North America's largest ichthyological collection, the Burke Museum of Natural History and Culture. But that collection really only goes back into the early 1900s. We get really lucky if we're able to find something from the 1880s. In contrast, the European museums will have specimens from like the late 1700s. So we get way further back into the past with the European collections. By comparing different world regions, you can also kind of compare the trajectory of parasite change in different regimes of human impact, right? Like the Mediterranean was super heavily fished, even by medieval times, right? By the year 1300. In contrast, the west coast of North America wasn't super heavily fished until after World War II, and Australia even later than that. So if you're looking to understand the imprint of human activities on parasite communities, looking at different world regions is really helpful because you can disentangle time from human impacts, knowing that that human impact happened at a different time in each location. So yeah, our long-term plan is to get into as many other world regions as possible, and we want to diversify the kinds of species and locations that we have data on.
Thank you. For our next question, we have Dr. Skylar Hopkins, which is, by the way, one of our panelists for tomorrow's panel discussion, asked, you talked a lot about how fishing can impact fish and their parasites. Are there other ways that changes to the ocean affect parasites?
Yeah, absolutely. One of the things that our team is working on right now is trying to understand how climate change influences parasite abundances in the oceans. You can imagine this going in either of two directions. Warmer temperatures should speed up parasite life cycles and give them basically more opportunities to reproduce in a given growing season, and you might expect that to increase their abundance. But also, parasites are just animals. They have a thermal envelope in which they're comfortable. You can make it too hot for them. And many parasites are adapted to cooler temperatures and might not do all that well as things warm up. So we really don't know how to make broad predictions about what's going to happen with marine parasitism in a warming ocean. Our team is trying to address this by working in the Gulf of Alaska. where different regions of the Gulf have warmed at different rates, with some remaining pretty stable while other regions have warmed up quite a bit. And by looking at museum specimens across a broad span of time from each of these regions, we can see whether the slowly warming regions have less parasitological change than the rapidly warming regions. And we can basically pull out the influence of climate on parasite transmission. We don't have any of those data yet, so I can't give you any answers, but that's how we're tackling it. The other thing that we think about a lot with respect to how ocean change influences parasites is pollution, both chemical pollution and nutrients. Again, we can come up with a bunch of different ways in which these different kinds of pollution might change parasite populations. On one hand, nutrients fuel phytoplankton blooms, which fuel copepod blooms, and copepods, these little zooplanktonic crustaceans, are key in a lot of parasite life cycles. So we might expect nutrient pollution to help parasites. On the other hand, putting too many nutrients into a marine environment can lead to this phenomenon called eutrophication, where you just get so much production that you can get low oxygen levels, and many animals find it difficult to persist in those conditions. So we can again see it going either way. We are trying to disentangle that in our Puget Sound data set because we have these amazing data on the nutrient pollution in Puget Sound from 1880 to the present day, and levels of various heavy metals in Puget Sound, including lead, copper, and arsenic. And we're going to be using our data set to try and understand how those various pollutants change parasite abundance through time. But again, you'll have to stay tuned for the answers to those questions.
Cool, thank you. For our next question, we have Sandra Avila asked, can you tell us if the prospective solution interfered with the analysis you performed on the samples? And if it was still parasite, or did you see the whole sample?
Mm-hmm. Yeah, so we were really concerned about this when we first started on this project because, of course, the chemicals that are used to preserve those fish are nasty chemicals. They're intended to keep fish tissue looking exactly the way that it did when the fish died. And we presumed that it would do the same to parasite tissue. But we didn't want to just rest on a presumption. We wanted to test that experimentally. So we do have a paper where we performed this experimental test. We got a bunch of fish and we randomly assigned some of them to be preserved using the protocol that museums use. Usually that involves dunking the fish in formalin to fix the tissues, and then doing a couple of rinses in water, and then some rinses in ethanol, and then storing in ethanol for the long term. So we did all those steps in collaboration with a couple of museum curator folks who are our collaborators on this project. And then for the other set of fish, the other randomly selected set, we just left them alone. We didn't do anything to them. And then we dissected all of those fish and we asked, are we detecting more parasites in one group or the other? And the answer is that we detect exactly the same number of parasites, whether you've got a fixed fish or a fish that's just been frozen the way that all fish are before they enter a museum collection. So we're very confident that the preserved fishes give us a complete picture of all the parasites that were in a fish at the time of its death. Now, does preservation change, for individual parasite species, our likelihood of detecting them? Probably there are a handful of parasite species that are a little bit harder to detect in the fixed specimens, but not so much so that we were able to detect any significant differences in that experimental study.
Thank you. And for our next question, Lacan das Singla asked, what can be the reasons for changing scenario in fish parasites?
Well, probably a bunch of things, right? The oceans are super different today than they were in the past. And one of the things that I really love about this research is just getting to sit down and try to time travel back 100 years and envision what the ocean might have been like at that time. We imagine that the ocean would have been significantly cooler because climate change has warmed things up quite a bit. We envision that it was significantly less intensively fished, particularly in the United States. Industrial fishing really picked up in the aftermath of World War II in 1945. So the oceans would have been far richer in hosts pre-1945 than they are today. And we also think that there probably have been changes in pollution profiles in the oceans over that time. Pollution's a little bit more complicated because it probably was increasing quite precipitously until the 1960s or the 1970s, when the Clean Water Act was introduced in the U.S., which led to a lot of restrictions on what could be released into ocean ecosystems. And so for nutrients and chemical pollutants, we usually see kind of like a rising trend until the 60s or 70s, and then kind of an improvement in water quality after that. Although, your mileage may vary depending on where you are in the world. So those are the big things that we think about. But different, regions might have other more specific, less global threats. For example, on coral reefs, one big impact can be anchors that are dragged by boats or damage to the reef structure itself. Not something that you worry about all that much in a rocky reef like what we have in Puget Sound, but something that's specific to coral reefs. So many, many changes, and all of them, we can envision many potential pathways for them to influence parasites. And that's part of why this research field is so exciting. There's very little known, which makes it quite easy to make pretty important discoveries.
Thank you. Then we have Mickey asked, do you have any thoughts on how invasive parasites or host jumping may change over a temporal scale?
Yeah, really good question. We work a little bit on invasive parasites. One project that I didn't have a chance to talk about that we are in our final year of at the moment addresses shell-boring polychaete parasites of oysters. These are worms that drill into oyster shells and set up shop inside those oyster shells. They're not particularly bad for the oyster unless they occur in really high densities, but they're super bad for oyster aquaculture industries because they create these ugly blisters on the inside of the oyster shell. And so when you're served an oyster on the half shell in a restaurant, you really don't want to see like a big nasty blister on the inside of that shell. They're not dangerous for people. They just kind of like ruin the aesthetics of the shell. And they're just so widely distributed. The most common invader is called Polydora Webster. It's thought to be native to Asia, but it's now in like every major oyster aquaculture region of the world. Until recently, we thought that the Pacific Northwest was totally free of it. We have this reputation for having these like beautiful pearly white oyster shells. But my team is always the bearer of bad news. And a couple of years ago, we discovered that those shell-boring polychaetes are here in Washington. They're also in Alaska. They're in Oregon. They're in Northern California. They're definitely here. And invasive parasites can be quite destructive. As Kevin Lafferty mentioned this a little bit in his talk. When you introduce a new parasite into a naive host population, it can tear through like that without the checks and balances that are normally on its population in its native range, and with hosts that have no resistance or evolutionary history with an invasive parasite. Invasive parasites can do quite well for themselves. So with increasing globalization, invasive parasites, of course, become more and more of an issue, not just for humans, but also for wildlife. And certainly over a long enough temporal scale, we should expect some global homogenization of parasite biodiversity just by dint of these invasive parasites being introduced into new regions.
And now let's start our Q&A session for Dr. Kevin Lafferty from the previous session, Parasite and Host Animals with Things. Okay, and hello, Kevin, thank you for joining us.
Yeah, I'm here, and great talk, Michael. It's such a, not only amazingly fascinating, but so important. I think one of the most important topics in parasitology today. So it's just so cool to see that.
Oh, thanks. I enjoyed yours. I wasn't at the beach for my talk, so I felt sad. My office was just a little bit more sad than your position.
Thank you. Okay, our very first question is from Sandra Avila, and she asked, I have only seen people eat cystic cerci. What did that fried parasite taste like?
Yeah, I mean, we really did eat those, and I would say it tasted like calamari, sort of sautéed in olive oil with a bit of garlic. Just everything you would want in a meal, basically.
Thank you. And for next question, still from Sandra. How did you solve the parasite problem in those Japanese fish?
So, if I understand the question, actually, it was my Japanese colleagues, Sato and all. They did this incredible field experiment, and I didn't have time to discuss it. But what they did is they went to these streams in Japan and they built huge cages to completely enclose long stretches of the stream to prevent crickets from jumping in the stream. And just to be sure that they kept all the crickets out, they put buckets of Kirin beer on the outside of the cages, because the crickets were attracted to the beer and they jump into the beer and drown. And so that way we were able to have sections of the stream where crickets were completely excluded, and other sections of the stream where the crickets were allowed to come in and go into the water. And that's how we were able to compare the changes in the stream food web. So it was really brilliant design that they did and managed to pull off.
Thank you. And our next question is from Hassan Chun. Dr. Lafferty, parasites are good and bad. However, some say complex life cycle parasites are better than simple life cycle parasites when talking about ecosystems. What do you think of it?
Well, certainly complex life cycle parasites provide more opportunities for the parasite to interact with the food web or with the environment. And so often the studies that we see that link changes in the environment to changes in parasites are based on complex life cycle parasites. That's not entirely true, but I think that's probably why people tend to make stronger predictions about complex life cycle parasites responding to the environment. In our system, for instance, many of our parasites, the trematodes we study, have 3 different hosts. And so if you change the abundance of any of those 3 host species, then you're likely to see a change in the parasite. So that's what makes them more sensitive.
Thank you. And our next question is from Eleanor. You talked about how the parasite coats the brain of the fish, but do we know what is the exact mechanism through which the parasites change the behavior of those fishes?
Yeah, it's one of my former PhD students, Denny Shaw, worked on that for her dissertation. And what she found was that the evidence suggests that the parasites are able to secrete a neuromodulator that has serotonin-like effects. And she did experiments where she had infected and uninfected fish that she had experimentally infected with the parasite. The parasites go in the brain, and then she would chase the fish around in a bucket and look at the stress response, which is measured through things like corticosteroids and things that the fish produces when they get stressed. And it turned out that the fish that were infected were a lot less stressed by being chased. So we know through just studies of human behavior modification by serotonin inhibitors and so forth that we can certainly manipulate human behavior by putting these powerful neuromodulators in the brain, and the parasite appears to be doing the same thing, and ironically making the fish more relaxed, which is probably a bad thing if a bird is chasing you and wants to eat you.
Thank you. And our next question is from Kyle. When studying coral reef system in the Palomar Atoll, did you factor in how pollution may have impacted the parasite communities? For instance, in World War II, this atoll was used by the US Navy, and recent research has shown that leftover pollutants remain in the concentrations that may affect the local ecology.
Yeah, that's right. We went to Palmyra not because it's necessarily pristine, but because it was not fished. And so the main comparison that we were able to make between Palmyra and Crisimas was the difference in fishing pressures. Palmyra does have some residual military contamination. It's difficult to detect. Like, you don't find it in the tissues of fish that they've looked at. So I'm not sure that it would be a big contributor to parasites there. However, in the heavily fish site, there's a lot of other pollutants, especially nutrient pollution that goes into the ocean there that can also affect the parasite life cycles there and might contribute to higher parasite loads in Chrysamis. And Chelsea Wood, she didn't talk about her work out in that area, but she actually looked at more detail and found a pretty strong effect of increased nutrients in places like Chrysamis that have increased some types of parasites, particularly trematodes that have snails that are grazers and so forth. So certainly, whenever we do comparisons and ask hypotheses about specific effects, it's very difficult to isolate just one single factor like fishing, because the environments differ in a lot of different ways. But at least from my guess is that contaminants at Palmyra Atoll were probably not playing a large role, but that's just my guess.
Thank you, Dr. Lafferty. And for the interest of time, I think this will be the end of our session. And thanks to all of our speakers today, and thank you all for joining us. Tomorrow, we will have our panel discussion, Parasite and Host Animals with Wings and Pouches, and our Parasites and Art section. Don't forget to join us again tomorrow for the second day of the event. And for tomorrow's event, we will be using a different link, which was sent to you by email through the Eventbrite. And a reminder email will also be sent out tomorrow with the YouTube link for the second day. So have a wonderful rest of your day, evening, or night. And we hope to see you again tomorrow at 6 p.m. Mountain Time. Bye!